Good morning. I want to thank the faculty and Professor San Nicola for his kind invitation. I'm going to present you with a lab study carried out in Liverpool on the delivery and transplantation of endothelium in DMAC with endothelium flapped in versus endothelium flapped out. At present, we heard that the delivery of the graft is done with endothelium out, and this is the shape of the graft in water. The endothelium flaps out, and this brings about the risk of damaging the cells during loading, during injecting, and during unfolding. So, based on the experience of DSAC, we wanted to see whether it would be possible to do it with the endo-in hypothesis. Is it possible to inject a DMAC endo-in, and which is the best way to deliver the DMAC graft without damaging the fragile endothelium, and how long does it take to perform surgery? This is a laboratory study, no patient, randomized, comparative and masked study on corneas that are unsuitable for transplantations from the Venice Eye Bank. DMAC preparation, stripping, the same for both groups, loading, then one using a cartridge, 2.2 cartridge, and another one, the other one, the Jones tube, and then delivery. And this is the video. This is the video. This is the endo-in. It means that DMAC, well, is already started, you know, stripping has already been made, and on the decimeric side, we do this threefold, as it was a DSAC, and then we load it on a 2.2 cartridge. The secret is using few drops of water just to help it slide in the cartridge. We take it and we put it in position. And you can control the orientation as well. And you see, the endothelium now tries to take the same position as it endo out. If it is done in the anterior chamber, it does it on its own. There is nothing to do, you know, it's easy, it goes, you know, by itself. And these are the outcomes. You see, this is the amount of cells grafted, endothelial cell density, mortality, and uncovered areas were measured. The uncovered area gives us the idea of the size of the graft, and endothelial cell density gives us an idea of the central part. The uncovered area tells us how many cells we have transplanted, which is the size of the graft that is covered with cells, and we also measured the time, loading time, injecting time, and unfolding time. So timing, no difference. Well, in terms of loading time, there is a difference between endo out and endo in, but with Venice Eye Bank we are working, we are trying to organize a project so that preloaded tissues can be obtained for a DMAC. So even if this time is longer with the endo-in, this time can be eliminated from surgery in a way, because it will be done at the Venice Eye Bank. The injecting time is more or less similar, but interesting is the unfolding time that is dramatically different. It is 1 to 2 minutes, you know, and this at present, there are not so many papers talking about the time and the amount of cells lost, and we do not know yet. But we think that the unfolding, the shorter the unfolding time, the more the cells we implant. In terms of endothelial cell density, there is no difference. No difference in mortality. There is a difference in terms of uncovered areas. There are more covered areas with the endo-in. In conclusion, of course, this study is only a lab study. We have to do it in patients. Endothelial density is not so different, but the only way to see the difference will be measuring graft survival in the long run, and that is why we are measuring uncovered areas, because it is the only way to understand what is the number of cells grafted. In terms of injecting and unfolding, there is no problem with the endo-in, and the unfolding time is really very short, and it is really significantly different as compared to endo-out. I want to thank the Venice Eye Bank with whom we are collaborating, and also our colleagues in Liverpool. Thank you.